Okay, I'm gonna kill a couple of seconds here because they usually somehow chop the first few seconds of my videos off when they go to load them on YouTube. Again, my name is George Paris. I'm a chemist, PhD chemist, dabbling in biology. Today is I guess it's the 20th of April, 2022. 2 days ago, one day ago, I don't know, I guess it was yesterday, I published a video and posted it on YouTube, and it's still there, uh, in which I was trying to summarize the evolutionary part, or not the evolutionary, the mechanistic part of the uh, what I call the Master Development Program. This is a concept that was published actually as early as 2009. I will mention that I have not really dealt with it, not really focused on it for about 5 or 6 years. The book, which I've mentioned a few times, this book was published. It's an Amazon publication. You can find it on Amazon.com. One of their self-published books, and I think it's dated August 2019. It relates back to a pre-publication. It was actually this is a paperback. The digital version was published in 2016, which was a bit more elaborate. And it actually went back to summarize papers which were published as early as 2009 in journals which no longer exist. For example, Biosciences Hypothesis doesn't exist anymore, published in 2009. Also published in 2010, 2011, 11, 13, and 14. So I haven't done any real work in this area for, around 10 years, about 8 years, I guess it is. And so frankly, I'm not as fluent in the technology, even the material I was working with, as I was 8 years ago. It's hard to pick it up. It's like, where was I? In any event, I made one mistake. I think it was either a pronunciation mistake or just the type of slip. I think in the last video I used the term proteasome when I should have used proteome. A little difference in pronunciation. Proteasome is a complex of enzymes which dissolves proteins, recycles proteins to their amino acids. Okay, proteome is the complex of all proteins in a cell at any particular time. So if you picked that up yesterday, 2 days ago, I think that would have been the 18th, then that's where that problem arose, and I apologize for it. There's probably other mistakes. These are relatively rough. I'm not doing this for money. It's not an effort to make a polished presentation. This is pretty much the way I would talk to students, so you just have to live with it. Anyway, after I got finished with that, I thought to myself, well, I haven't looked at the literature for 5-6 years at least. Haven't done anything serious. So I went back and I picked up a citation, one of the ones which I was using in the books and other things. I think the guy's name was Shepelev. I just searched that guy's name, and out popped, remarkably, a series of 8 papers. Let me show you what I got here. Let's see if I can get that onto the screen. This is in PubMed. I need to get the light on it and whatnot. There are 8 papers that's circled in red there, and they are all published in 2021 and 2022. And lo and behold, one of the points I made in the video was that as of the time that I was working on this, nobody really knew what was in the pericentromeric heterochromatin. These guys have now sorted that out. Now, to put this in perspective, this is when I published my papers. You'll notice there was 3 of them published in 2009 and 2010. That actually made it into the PubMed group. Interesting story about how that happened. Like I said, I originally published in a journal that no longer exists. When that happened, it's like, oh my God! When that journal disappeared, it was like, oh my God! You know, what am I going to do? Because that's they actually had the copyright to the material. And so I went to Medical Hypothesis, friendly editors there, and they allowed me to recapitulate, bad term, I guess, reestablish or restructure the idea into a medically oriented paper, which is actually what got published there. That was 2009, 2010 type time frame. So this, the fact that this popped up, these guys have done amazing work. There's about 50 authors on one of the papers, over 50 authors on one of the papers. There's several other papers that have other authors. I don't know any of them. There's a tremendous amount of work going on. Has been done. I knew that was happening. I knew that things were moving fast. I didn't realize that literally those papers got published. I think it's on April 1st. This is April 20th. It got published online about 3 weeks ago, April 1st of 2022. And I'm very astounded by the work. You're looking at 10 years of intense work by literally probably hundreds of people. Obviously, I haven't gone back and read all that. I don't plan to. I'm at a different point in my career. I'm 75 years old, and I've been, for the last 5 or 6 years, I've been actually looking at historical materials and published some historical documents. And currently, I'm actually started a company with a friend of mine, Directed Energy Materials, LLC. And I'm actually working with a collaborator at the University of Maryland, medical centers, and we're doing some research there. And this is really occupying my time. So I have no intention of going back and trying to become competent in this material. It's way beyond me now. It's like these guys, there's years and years and years of research that are far beyond what I've done. That's one of the reasons I point out that this is a hypothesis. These guys they may already know that I'm totally screwed up. Hopefully, if they figure this out, they'll come back and say, "Hey, Dr. Pearce, you have a problem. Forget about your idea. It's something else that's happened." That may be the truth. So, if it's not what happens, if they come back and say, "Well, hey, we're working on this," I'd say, "Very good. That's what I want you to do." In any event, what I wanted to do, I'm going to continue doing what I started doing. What I promised you I was going to do was to talk about the evolutionary part of the hypothesis. In other words, when I did the hypothesis, I was purely trying to design a system that was chemically plausible, that would explain what I observe, what I understand to be human development, or development of higher organisms. I did not at that time focus on evolution, although I was interested in evolution, but I didn't anticipate that the hypothesis would explain evolution. It turns out, I believe, that the hypothesis with supplemental material does explain evolution. That's sort of where I want to go today. To get into that, I want to go back and talk about evolution a bit. Some of my earlier videos in the series, if you watch them, you may not need to watch the next 15 minutes of this video, because that's what I'm going to do. Is I'm going to go back and mention a fellow by the name of Charles Darwin and his colleagues. And as you recall, Darwin's introduction in this area was strongly impacted by the fact that there was a religious concept of creation and how we all got here. And Darwin was basically punching through that with a scientific approach. He and Russell Wallace. Okay. So Darwin had, now I mentioned two people that were associated with Darwin directly. Huxley, Darwin's idea was that evolution leads to speciation, but it takes a very long time. In Darwin's view, it's a very slow, progressive process. Okay. Huxley basically said, hey, you might want to soft pedal the idea that it takes a long time. It might not take very long. If, you know, in some situations, you might have relatively quick speciation with very little evolution in between. Darwin's basically, no, don't want to go that way because of, you know, his interpretation of facts. Along came a guy named Ernst Haeckel. Haeckel was more of an embryologist than anything else, and he was fully absorbed by Darwin's idea. And Haeckel, through looking at the embryos of various organisms, came to the idea that our development of our embryos, that is to say high-level primates, humans, etc., embryos, are reflective of what had gone before. The idea was that our embryos represented variations on the previous materials, which is pretty consistent with Darwin's idea. The mainstay of what Haeckel did was to publish a series of graphics. These were hand-drawn pictures that he had created from his microscopic views of these things, which represented embryos of high-level mammals back to, you know, back to amphibians and that sort of thing. Now, on the one hand, we know that we're basically looking at that we all start as single cells and we go to these ways. So he basically felt that he could fill in the blanks. Now, I think the idea was that ontogeny, that is the development of one particular organism, like me, reflects all the phylology, that is to say, all the development of the phylum that I belong to, the species I belong to. He carried that to the extreme, and to assist people in seeing what he was interpreting, he tended to fake his drawings a little bit. Okay, he emphasized things that he thought were consistent and sort of ignored some of the things that were not so consistent. As a result, people didn't pick, well, some of his peers understood that there were some discrepancies here, but his message and Darwin's idea was so powerful that throughout the early 1900s, Haeckel was like 1866, and this phone is going to drive me crazy. I'm going to kill this people. In my age group, I get a phone call about 3 times a day telling me I should to go get vaccinated. That's just a little after the mask mandate went away. I believe I haven't been wearing a mask for a long time. I did have COVID a long time ago, but I survived. And then I had to get vaccinated. And my foot is tangled up in the cord. I apologize for this nonsense. There we go. I'm back. So anyway, going back to Haeckel. Haeckel dominated early 20th century, early 1900s biology text. And as late as the 1950s, probably 60s, I was taught, and everybody else was taught, that that's the way it was. That was the signature support for the Darwin theory. Well, in the 50s, 60s, there was a revival of religious views with the idea that intelligent design by a creator would account for this, and that we should sort of forget about some of the Darwinian ideas, and certainly Haeckel. Well, what happened was Haeckel, in order to avoid this issue, the Darwinians, most scientists, basically threw Haeckel under the bus. I mean, it's like, this guy's a total fraud. Out. That's not quite true. Haeckel has some insights that were valuable. The problem I see with the Haeckel idea is that suppose you have humans and you have, say, fish, okay, or frogs or something like that. Okay, there's no real reason why a current day evolution of the fish, that the embryo that precedes the fish, should look anything like the embryo that preceded me. Okay, why? Because there was a lot of evolution of the fish and me from our common ancestors. I believe we have a common ancestor, but by the time we both evolved over millions of years, why would we expect the embryo of the fish to look like the embryo of the human? There might be some major characteristics, some very fundamental issues that go in there. And I think perhaps they are reflected in the embryos. In other words, there's stuff that preceded both of us that end up in that. But it's, I would not expect there to be a great correlation. There should be some correlation, but not great. We get into an area where we're basically quibbling about how much, and there's no complete answer to that. So anyway, Haeckel's theories are now pretty much not appreciated in science, primarily because it opens up the criticism that the religious groups, the intelligent design people have against the Darwinian approach. All right. Now Darwin had his, I mean, there are problems with Darwin, in my view. Darwin had the idea that, as I said, that evolution leads to speciation. The problem with that is that nobody really understood what is a species. The concept of species was in the 1800s mainly dominated by people who do, I guess, the taxonomy on fossils and things like that, how the fossils looked and how they changed and all that sort of thing. And so they would say this one is different than that one, they're different species. I think that's kind of wrong. The idea that if you start trying to try to apply Darwin to modern genetic and genomic understandings, and this is developed through the Morgan group, and which included, I guess, Mueller and his artificial mutations, etc., 1930s time frame. The issue that comes through there is that how do you get this variation that would be needed for the natural selection to occur? This is where everybody got focused on the proteins because that's what they could study. And I think most evolutionary biologists look at Darwin and basically say, okay, you have this population, and in order to have the population go to speciation, how does it change so much that it's a different species? The idea was, okay, there must be some sort of physical barrier, like a mountain range or an ocean or something, that separates these two populations of the same species, and so that they go out and they evolve separately. And when that happens, and eventually they become different species. Well, we have a number of examples where we do have populations that get separated with physical barriers. You can look, for example, there's a salamander in California. I think it started in the north of California, and one took the coastal route, one took the mountain route, and they've got down to Southern California now, and they look quite different. They don't look like the same species now, but they still can interbreed. Okay, then you have Darwin's finches. Darwin's famous finches from the Galapagos. They look different. They have different ecological niches. Some eat seeds, some eat insects, whatever. Different beaks and things that if you were doing taxonomy, you would say they're different species. And indeed, he felt there were different species. But recent observations say these organisms can interbreed. They have taken on these different niches because that's convenient for those groups to survive. We can look, these are called, by the way, so these are called, some of them are called ring species. You start out here, you go around, and then come back together and it's all the same species. You can even look at humans. Okay, you have your Africans and you have your Europeans and you have your Asians. And we look superficially different. But guess what? We all managed to converge on North America and we're all interbreeding very nicely. It happens quite well. So these organisms that look different are not different species. I will say that in the 1800s there was a very big debate, very serious debate among the most erudite people at major colleges, universities in Europe and in North America as to whether or not Africans and Europeans were in the same species. Okay, this issue about what a species is, I don't think has been resolved until Ernst Mayr. Ernst Mayr is a guy who lived a long time, and in 1942, Ernst Mayr came up with the idea that a biological species should be defined by those organisms that can are mutually fertile. In other words, if two individuals can get together, produce offspring, and those offspring are fertile, then you have a species. If it can't do that, then you don't have a species. All right. So I think that is a objective, clear-cut definition of what a species is. It takes out all of our prejudices and biases about what things look like. Now, concurrently with that, and this was an issue, a fellow by the name of Richard Goldschmidt, in the 1940s, back perhaps a little bit concurrent with or slightly before maybe even Mayr, said, hey, he was sort of like looking back at Huxley and saying, hey, you could conceivably have a massive mutation. And if you looked at, for example, the work of Mueller, where he had really large mutations in the genome of things that he shot with x-rays and whatnot. If you had a really major mutation, you could get a monster, a completely different looking organism, which might be viable on its own and might found a species. Well, the counter argument to that, which has been used in many biology classes since the 1940s up until the 2000s, was it, well, how does this hopeful monster find a mate? It's like the idea that two people or two of these spontaneous variations would occur out there and could come together and make a new species is very unlikely. I think it's totally unlikely. But when I got to thinking about the pericentromeric heterochromatin and read into this, it became apparent to me that there's a major mutation, namely the inversion of the centromere, that not only doesn't really change morphology, but it does inhibit reproduction. And there's no reason why you couldn't have two people like that could be fertile, mutually fertile, and could create a nascent new species. Now, what do I mean by all that? If this process, by the way, is called saltation, it's one generation to a new species. Okay, and it's something which flies in the face of Darwinian gradualism. I published a paper in, I guess it was 2011, which summarized this. Okay, this is one of the defunct journals that I published in. Okay. This happens to be a hypothesis in the life sciences, which at one time was sort of a spin-off of medical hypotheses. But in any event, it occurred to me after, actually, I think it may have occurred concurrent with or after I read this book, Flux of Eden. I love this book. It's a discussion of evolution and human genome. But one of the things that became apparent in this is if you look at modern chimpanzees, I'm trying to find a place to put this without losing it entirely. If you look at modern chimpanzees and gorilla and humans, what you find is that there are identifiable inversions. I think on the order of, I think humans have like, if you look back to the common ancestor of humans and chimps, you can deduce that there are, I think it's 7 inversions in the human line, and there are, I think, 9 in the chimpanzee line. And it's more as you go, further back to the gorilla and pongo, the orangutans. This is interesting to me because the normal population of humans, which is now billions of people, have all the same inversions. How did that happen? The only way I can think of that could happen is if the inversion was concurrent with speciation, and indeed, most likely, was the cause of speciation. How are you going to explain that otherwise? All the chimps have the same set, different from ours. We have a set different than theirs. That is to say, the standard model of a human and the standard model of the chimp, that represents 99% roughly of the two species, have, 99 out of 100 humans have the same set of inversions. 99 out of 100 chimps have the same set of inversions. So how did that happen? Unless the inversion actually happened at the speciation event, was the speciation event. I think that's very strong evidence that the inversions were the speciation event. It has nothing to do with change in morphology. It has everything to do with changing in the reproductive success. Now, in my paper, I showed you a minute ago, I talk about, there's an example in mice that's kind of interesting. It's not a pericentric inversion, but there's an inversion out there that seems to make the mice no longer function. There's some examples in horses that seem to be tied to this, and there are, in fact, changes in humans. Now, how do we get the 2 people with the same inversion at the same time? Well, it's fairly simple. You could have a spontaneous inversion and pass it on to more than one child. Or you could, that could be passed on to first cousins, etc. There's a taboo in Western society against what we call incest, having sex with local close relatives. But first cousins are pretty close. They're close enough that this becomes a very likely possibility. And Darwin married his own cousin, okay? And I think in most states in the United States, it is not illegal to marry your first cousin. Some states have various variations on that. But first cousins are clearly within range of one another and could clearly receive the same inversion from a grandparent. And so this idea that first cousins could get together, create a nascent species, that is to say, a few individuals who were not able to interbreed with other people, and that it's well known that these inversions cause reproductive failure. When you try to breed one person with an inversion with somebody who doesn't have that inversion, you have a problem. And so it's quite possible that you could have these nascent species. Now, it doesn't, in my mind, it doesn't even necessarily require that you have exactly the same inversion. That's to say, right, exactly the same cleavage point. You might be able to have slight variations in that which would still work, which basically opens it up a lot. Now, it turns out that, I think I mentioned a couple of days ago, that the inversions, in humans, it's chromosome 9, has about 1% inversions in chromosome 9. That is, 1% of the population has an inversion in chromosome 9. That's pure centromeric inversion. And so that's, when you're talking about billions of people, that's a lot of people. So we probably have nascent species of humans running around the planet right now. Now, they are kept together by, among other things, reproductive essentiality. In other words, if you want to have children, you find out you can have children with your first cousin, you probably hang in there, and you probably have these groups of people who are spreading as a small population, as a separate species. And given ecological differences or ecological changes, they may suddenly find themselves to be the favored species, and it could cause a tremendous expansion in their population. They could become the dominant species. Now, obviously, in the old thousands of years ago, the human population was much, much smaller. You go back 20,000 years, we're talking much, much smaller populations, and there were fewer people to choose from. Today, you go on the internet, you start finding thousands of possible mates. But a few hundred years ago, you basically married some girl in your some girl or some guy in your local community, because that's the ones you met. That's the ones that were there. Okay. So things have changed a lot, and so the outbreeding opportunities are much larger than they were many years ago. In any event, so I think that explains speciation. And let's go forward from there and talk about how the Master Development Program can be enhanced to incorporate the variation needed for evolution. In other words, evolution, I believe in Darwin's idea of natural selection, which really wasn't original with him, but natural selection based on the ability to survive under adverse conditions, etc., favored species, etc., makes perfect sense. Now, if you go to some more modern biological thinkers, I'm thinking about Stephen Jay Gould and Niles Eldredge. They came up with two ideas. One was punctuated equilibrium. Now, this is an observation in biology. This is not a hypothetical thing. This is something that they observed and led them to this idea. Well, what they observed was that certain species seem to persist for millions of years without changing significantly. And presumably, over those millions of years, there were some variations in the environment which would have preferred something. But suddenly, boom, occasionally you would just have a new species arrive in the fossil record that had not been there before. There was no evidence of a transition, a Darwinian evolutionary tree. You didn't have this type of relationship. You had a completely series of parallels. It's like we first had this one, now we got this one and that one, now we got three others, and they just sort of appeared in the fossil record. And this was puzzling to a lot of people, like, how can you have that? And these guys basically came up with the idea that there's a stasis, that species don't sort of meander away. They just tend to stay the same regardless of what's happening around them. And I think that's perfectly correct. Okay. And because in my view, if you're having sort of random variations, it probably stays very close to the norm, whatever's favorable. And so this idea of a punctuated equilibrium, where you have equilibrium and then suddenly you have something new happening, that's actually explainable by the ideas that I'm about to present you. Another concept that they introduced was terminal addition. And this would go back to Haeckel. Basically, if you know, in my mind, if you're looking at random mutations, this is the argument against Darwin. This is the intelligent design argument against Darwin. If you have random mutations, they're just arbitrarily popping into the genome, the odds of coming up with a genome like ours is very, very small. And in particular, the pathway to it would be chaotic. It would be, you know, like one day we looked like a fish, the next day we looked like a lizard, the next generation we looked like a chimpanzee, and then we looked like a fish again, because of, you know, the randomness of this whole process. Now, terminal addition, which is essential for this idea of ontogeny recapitulating phylology, basically says we're building on something, we're adding to something. We start off with this, but we can add to it and, you know, go away. So we can start off with a single cell, like some protist, and we can add to that and get to something new and something different. We can go to a multi-cell organism. We can start looking like a sponge, or we can start looking like a plant, or whatever. We can start looking like a starfish or shark or whatever. You know. So we can go through all of these different phyla by addition, and that goes back to Haeckel. That goes back to ontogeny recapitulates phylology. And I think that at that level, if you were looking at the embryo of the fish at the time that it departed, or that the time that our embryos departed, you probably would see more correlation with our embryo. But we can't go back and get that embryo, because that embryo is tens of millions of years ago. Okay, and it's gone. What we have today is that embryo after it has evolved for millions of years. And so we shouldn't really expect it to look like the embryo of a modern human. Okay, it doesn't. You know, the modern fish is not the precursor of the modern human. We share some earlier common ancestor. So anyway, I'll throw out those two names, Niles Eldredge and Stephen Jay Gould. If you're not familiar with them, you should be. And now let's go back to my hypothesis. The way I see this working, remember, for mechanical reasons, purely mechanical reasons, when the chromosomes are dragged apart, there is tension in those strands, and they are susceptible to breakage. The heterochromatin is multiple strands. It's like having a rope that's a very thick rope, and it's mechanically stronger than the places where it's joined. It's joined at the centromere, and it's joined at between the pericentromeric material and the euchromatin. And they're prone to be breakage at those two spots. And when that breakage occurs, that breakage is manifest, among other things, in these pericentromeric inversions that I've been talking about. And so what we see, and this is what I think there were 2 or 3 papers when I started writing this that said, hey, there's this breakage, and that, you know, your pericentromeric material grows from the center out. In other words, the most recent stuff is near the center, and then you have the older stuff further out. There were only 2 or 3 papers when I said that. Now there's like 8 very strong papers that talk about in great detail this. So that's one of the reasons for jumping right on this and going with it. There's much more evidence about what's going on, how it went on, etc. And one of the things that's noted is that a lot of this, what's in there is the DNA are actually retro transcripts of RNA, which were the result of retroviruses. Somewhere I read recently, and I'm not sure where it was, in the, say, 50 million years that we go back since the dinosaurs, roughly, there's been something like 31 retrovirus pandemics in human history in the last 50 billion years. So that would be like once every million years or so you get a pandemic of a retrovirus. Right now we have HIV, I guess, is a retrovirus. And so these retrovirus segments get embedded in that, when you have the break between the centromere and the pericentromeric material, in falls a retrotranscript, that is, say, a piece of DNA that was transcribed from a piece of RNA of the virus gets stuck in there, and we go with that, and that becomes fodder for new variations. Now, along with that material, we have a lot of other stuff. There's in the normal cell you have all kinds of transcriptions going on all the time, and this would be true of the gene cells as well as the somatic cells. Somatic cells don't really matter. Okay, it's the gene cells that are important for evolution. So you get these breaks. You have transcription going on, reverse transcription going on. You have all sorts of pieces of the gene, many of which don't end up in the final coding for the final protein. They are untranscribed areas, and they are not untranslated areas, and you have introns, etc., which are popping out of there as the original code is prepared for export into the cytoplasm. And these can be retrotranscripted and end back up into the genome, along with the retroviruses. And then of course you have all the usual stuff that can happen once you have a piece of DNA embedded in the heterochromatin. So I have this little drawing here. This appears in the book, which basically summarizes what I'm talking about. So this is how the hypothetical master development program can incorporate variation into the genome. Basically, you have transcripts of genes, lines, signs, etc. You have the retrovirus, and during a period of retroviral infection, you can have a very substantial amount of material generated, which ends up in that break point between the centromere and the pericentromeric part of the genome. Now, you've noticed, if you sit there and look carefully, these generation-specific control keys, GSCKs, start off with 1 at the right side of your screen, and then there's 2, and then there's N-1, and N, etc. Okay, so they're growing. The most recent ones are coming from that gap between the pericentromeric heterochromatin and the centromere. All right, but there's other variation happening out near the euchromatin. So-called seeding and swapping with other chromosomes are happening out there. So I guess I'll toss this one out next. Hope you can read that. Get some light on the situation. So basically, you can have all these sources of RNA getting converted into pieces of DNA. Those pieces of DNA, miscellaneous DNA transcripts, getting dropped into these breaks between the euchromatin and the pericentromeric material, and between the pericentromeric heterochromatin and the centromere. And then you have variations occurring, seeding and swapping, and natural selection going on. And I'm envisioning that if these are in fact the generation-specific control keys in their various forms, then the execution of this would probably be from the outside, let us say the infant, towards the center, towards the centromere, through that sequence. So that's how I'm envisioning variation being introduced into the genome by this process. Now, if I summarize the whole process, this gets very busy. In my books, I have copies of these things which you can take and study if you're interested in it. And I apologize, it looks like I sort of got off the thing. Here I sort of map out all the various things that are happening here. And I'll sort of scan back and forth through it. But you basically have both of these processes. In other words, the development processes are going on and the evolution processes are going on at the same time. And the development processes are particularly important when you have retroviruses. So basically, you've got intron, secondary activation, cascade, etc. By the way, I think these introns that are very important from the standpoint of coordinating what I call the delocalized operons. I think that they're not there just for harassment. I think they actually contribute to the operation of the operons. And so you get this execution towards the centromere. Down at the bottom here, this bottom box is what's going on in the cytoplasm. One of the things I haven't really emphasized, this so-called translation of an initiator protein, that is not a unsupported step. Although the use of that protein to actually seek out and activate the specific generation-specific control key is a novel idea. It is well known. If you go look, there's papers dating from about 1988. The authors are Blow and Lasky. Blow's got hundreds of papers published. Basically, it's well known that there's a protein that is coded, transcribed in the fairly early phases of the mitosis process. It ends up in the cytoplasm. It's transcribed it's translated in the cytoplasm into a protein. That protein is prevented from getting to the nucleus by the nuclear membrane. And it's only when the nuclear membrane breaks down that the protein can get back into the nucleus and kick off the next cell cycle. Look up Blow and Lasky, 1988. That's an important paper. I'm envisioning something like that happening, but more specifically, not only does it kick off a cell cycle, but it also selects what the generation-specific control key might be. Now, that could be a default factor because the previous ones may be completely shut down and just the next one that's available, or it may be a specific control key. I can't predict that. So anyway, you have these things going on, and let's look at how that impacts evolution. This drawing also appears in my book. And what I'm envisioning here, let's go down to the bottom here. At the bottom, I have indicated some environmental parameter. That environmental parameter could be oxygen content of the atmosphere or the water. It could be salinity. It could be UV radiation. It could be any of a variety of possible things. It could be atmospheric pressure. It could be, availability of a plant species for nutrients, etc. Okay, so that variable is there, and it changes. That's the dashed dot line that's in there. So I'm just arbitrarily saying it goes back and forth. Well, I envision this original species. I'm just saying that when that original species comes onto the screen, it happens to be perfectly aligned with the environmental variable. You'll notice at the bottom we have everything together right here. But the environmental variable changes, whereas the original species doesn't change. It's in stasis. We're talking about a species that continues on for some time. Now, at some point, there's a retroviral pandemic. I guess that's this process right here. That retrovirus pandemic creates a new species, which happens to be able to respond to the existing new environment. That is to say, the environmental parameter has changed, and it's able to flourish there for a while, but it meets its demise when that environmental parameter changes back. It becomes extinct in my little diagram. And then we go up a while longer. We go through some oscillations, and finally the environmental parameter takes a real whiplash, and the original species is no longer viable. But luckily, there has a retroviral pandemic about around that time, and a new species was created, and that new species, we call it species B, is able to survive under the new conditions. Now, species B is probably going to get wiped out when the environmental parameter changes again, but that's the way life is sometimes. Hopefully, if for the continuation of this line, hopefully there will be an opportunity for there to be a new species, A type. So that's how I envision that's all happening. And going back to this issue of what can be called the phenotypic stages, if you had sort of random evolution, you'd expect these stages to pop off randomly through this process. And you'd expect if you plot development on your abscissa and evolution on your ordinate, that's what you would sort of get. And it's the observation is that the phylotypic stages don't go through. They're not. They don't look like this. Something else is happening. And I mentioned earlier in my last presentation that one of the things happening is that the fetus has to evolve with the adult, such that, and that's accomplished by the fact that you have terminal addition at both ends of the generation-specific control keys. And you go through something like this where all the different fetuses, all the different immature versions have to converge to a specific phylotypic stage and then progress from there, because there has to be evolution of the early stages along with the late stages. The adult and the infant have to be consistent in order for everything to work. And so the development program which I proposed is consistent with that, which is kind of cool. So anyway, I'm hoping that out of this flurry of activity that we are now seeing from these folks who have managed to sequence pretty much the entire genome, that's these guys again, which all happened within literally the publications were the first of this month, like 3 weeks ago, 2022. Hopefully they will explain or either confirm or deny the hypothesis which I put together back in 2009, okay, and haven't done anything with it since 2010. All right, so I'm not in that ball game. I don't plan to be in that ball game. I don't have time or energy to pursue that. I don't have academic credentials to pursue that. So I'm going to be happy being a chemist. I'm doing chemistry these days. And as an old person, like I said, I've started looking at history a bit. I published some history books. Most everything I've written is on Amazon.com. You can find George E. Paris. There are other George Parises out there. This Paris is a fairly well-known name, turns out. And I've been fascinated with genealogy and the genealogical connections by the people who sequence these things. I've been sequenced. I'm in the family tree database, and I guess I'm on GEDCOM, whatever it is. In any event, so I think this is an interesting area. I think that it's a phenomenal success by these guys and gals, or whatever pronouns happen to be. These people who have successfully transcribed T to T, T to T, that's like B to B, T to T, which is telomere to telomere, these chromosomes. That's the new buzzword, I guess, T to T. In fact, that's how you can find this in PubMed by looking for T2T, and I think it's CEM13. I'm not sure what the CEM13 totally refers to, but T2T probably will get it as well. I will be very interested in seeing what these guys and gals, people, develop, and I wish them great luck. And I would, whether, as I said 2 days ago when I did the previous video, I look forward to either being affirmed or rejected, one or the other. Okay, take care. Have a great day. Today is the 20th of April, 2022, and I really could use some of that global warming stuff because it's been cold here in Gaithersburg, Maryland for the last 3 or 4 weeks. It's been unseasonably cold. I had to go out and cover up all my flowers to keep them from being killed by frost last night. So now 2 weeks from now I'll be cutting grass up to my neck. Have a great day, and bye from here.